Hello there, and welcome to Teach Me in 10. Have you ever wondered how cutting edge drug development can be accelerated? Well, today's your lucky day because we'll be diving into the game changing world of screening strategies with Peter, who's an expert marketing product manager from ACROBiosystems. He'll reveal how these innovative strategies are not only transforming drug development, but also paving the way for breakthroughs in medicine. So, without further ado, I'll hand over to Peter.
Hello, my name is Peter Hsueh. I'm the marketing product manager at ACROBiosystems. It's my pleasure to introduce you the title of my talk, Screening in Drug Discovery, in this Teach Me in 10 webinar hosted by Technology Networks. When we look at overall drug discovery and development workflow, we identify there's a very lengthy and complicated and costly process, starting target identifications, to assay developments, to molecule library screening, initial screening, effectiveness and safety assessment. If you're lucky, they've done all of that, leading to preclinical study and clinical trial eventually for commercialization. So this is during the early discovery and this development process, we identify the key step, including initial screening and molecular library screening, can be shortened if you have a better understanding of the technology that are available to you. So screening is important, right? So we understand it's a critical step in the whole drug discovery. Identify drug candidates that start by failing to understanding specific biological activity, pharmacological effect, and potential therapeutic value can help facilitate the whole process. Because you look at the data below, there's only 1 to 2 drugs actually make it to the market out of 1,000 leading candidates. So it's a very small percentage of drugs actually lead to the market and become more and more stringent. So during the discovery and development stage, we really need to understand, we find the advantage of a different drug screening, including uncover novel therapeutic targets, reveal the high potential drug candidate, to help accelerate the whole drug discovery and development process. Here to simplify the goal for drug screening. We want to improve the speed and efficacy of the drug discovery developments. Right? So by identifying the potential molecule faster, shorten the drug development cycle, and reducing the drug development costs. And with that, hopefully they are leading to faster and better drug discovery to help benefit the patient down the road. There's a lot of different tools and platforms available to our researcher now for drug screening, based on especially focus on target drug binding evaluations. So first is the drug library constructions. Library may include nature products, synthetic compounds, and different biologics, including antibody. Specifically for antibody library, different methods such as hybridoma technology, manual, and phage display can be employed. So next is the high throughput screening. Methods such as fluorescence-based assay, radio labeling, and affinity chromatography, and more are commonly used in high throughput screening. Adjacent to high throughput screening is the target drug binding analysis. Binding and affinity can be measured using traditional techniques like SPR, BLI, and ITC, and fluorescence polarizations. Also, cross-reactivity can be used to validate the specificity of the candidate. Last but not least is functional validations. To demonstrate the drug candidate modulate the function of the target in a designed manner. This kind of evaluation can be done by cell-based assay, 2D, 3D cell culture, also introduce organoids, animal model, and biochemical assay as well. So we talk about conventional SPR being one of the binding affinity evaluation typically used by drug developer, but they have some difficulties, especially in throughput, make it hard to use and expensive to screen for a lot of biologics candidate like monoclonal antibodies. So antigen binding make each experiment longer and introduce potential error in reproducibility. Here we'd like to propose our alternative methods like high-throughput SPR by collaborating with Cutera. So high-throughput SPR is a powerful and robust technique that explores drug candidate against different format of transmembrane protein or hard express protein like Claudin 18.2, CCR5, with stable receptor that such as nanodisk or VLP and detergent myocyte provided by ACROBiosystems, give us more confidence in the report of binding measurements and reproducibility. This allows 100 monoclonal antibody being screened in parallel to reduce the cost of labor, reagent, and time by using high-throughput SPR. You can see on the right. Next, I also like to mention functional validations using organoids. So organoid is a 3D miniature model aimed to recapitulate the specific characteristics and functionality of an organ you look for modeling. There are different ways to culture and grow organoids. At ACRO, we focus on provide iPSC-derived organoids. There's a few key advantages, including a better consistency with genomic and transcriptomic stability from one donor, and also that's aimed to have a consistent drug result in comparison. And also have a larger production capacity, allow high throughput screening down the road with the complexity, necessary complexity in human genomic background. So a lot of labs have been adapting organoid in vitro to study disease model in their research, and they have been leading to translatable result by using human iPSC-derived organoid to fast-track preclinical evaluations and studies, specifically for more assessment in efficacy and safety for the drugs. Next, I'd like to introduce you a couple case study and applications using our organoids. First, looking at the cerebral organoids to screening AAV serotypes. So AAV are critical in gene therapy as a main delivery system for genomic payload. So it's important to screen the appropriate AAV serotype to specific organs or specific cell type to critically ensure the high gene delivery efficiency to help you increase the on-target effect, also result in higher gene expressions. So you can see the data here. IVB2, a modified serotype related to AAV5 wild type that had increased infectiousness toward the cerebral organoids that make a positive result. You can see IVB2 compared to wild type have a better success and the potential treating for neurological disorder. Next, I'd like to introduce the second organoid applications. You're looking at cardiac organoids, specifically in the context of drug toxicity evaluations. You can see in the control stained by DAPI for nucleus, CD31 are standing for endothelial networks for vasculature cells, and also CTNT is marker for cardiomyocyte. You can see when they merge, it's a fully functional cardiac organoid with a defined structure. But when we introduce a couple drugs, including drug A, that seems to toxic mainly specifically to the CD31 positive cells that disrupting the vasculature network. In contrast to drug B, both seem to be disrupting the CD31 positive cells, also the CTNT positive cell that can be leading to very interesting evaluation when you look at the mechanism of the specific drugs. So here are two examples for using organoid to uncover more insight about your specific drug in terms of toxicity, also AAV serotype. In addition, we have a lot of variety of different solutions for drug screening, including our protein products, in transmembrane protein, like I mentioned, immunocheckpoint protein, also Fc receptor, inhibitory screening kits, and other products. Here's a whole summary of different product and service that we would provide for drug discovery researcher. So if you're interested to learn more, feel free to go to ACROBiosystems.com to learn more. With that, thank you for listening to my talk. Hopefully you learned something, and best luck with your journey in the drug discovery.
We're so grateful for ACROBio's support in bringing you this episode. They're leaders in recombinant proteins, kits, antibodies, and scientific services, known for their high-quality standards and support in biopharmaceutical R&D and clinical applications. So if you're curious to learn more, you can click the links that we've left in the details for you. Thanks so much for tuning in, and we hope to see you again soon for another episode of Teach Me in 10.